Now let's learn about the culture methods used in virology. These can be divided into three parts. One is in animals, one is in eggs, and one is in tissue itself. So let's focus on the animal part first of all. Animal inoculation. The most common organism used is mouse, and for Coxsackie virus and arboviruses, the mouse used should be a suckling mouse. Second thing is egg. The egg usually is approximately a 10-day-old egg and embryonated egg. It will have an embryo over here, and as you can see the diagram is very important. This red colored outline is a shell, the egg shell, and these broken lines, these form the shell membrane, and this part is the air sac. All right, and this red part, this is allantoic cavity, which is formed by a membrane known as CAM, chorioallantoic membrane. And this one is embryo surrounded by a dotted line, amniotic cavity, and this dark colored dot like structure, or this one is yolk sac. And it is important to learn that which organisms can be grown in which part of the egg. Okay, allantoic cavity, this red colored cavity, what can grow in it? Yellow fever virus, rabies virus, and influenza virus. Yari, YARI, yari can grow in allantoic cavity. Allah में yari होती है. Yellow, rabies, influenza. And this embryo has amniotic cavity around it. Amniotic cavity, myxoviruses can be grown over here, influenza viruses. And yolk sac, this dark structure, yolk sac can grow 2 bacteria, chlamydia and rickettsia, and 2 viruses, Japanese encephalitis and West Nile fever virus. So yolk sac, 2 words, yolk sac, 2 viruses and 2 bacteria. And the CAM, the chorioallantoic membrane, pox viruses grow on it, and molluscum is not included. It does not grow over here. Good Pasteur was the person who first used the hen's egg for viral cultivation. Now let's focus on tissue culture methods, which are very important. Divided into 3 parts: organ culture, explant culture, cell line culture. Organ culture, the organ or the tissue outline and architecture is not destroyed at all. Like nowadays coronavirus cases are there. Tracheal ring culture can be done for growing coronavirus. Explant tissue is destroyed, architecture is destroyed. It is rarely done now. Most common method is cell lines. Cell lines, what happens in it? The tissue with the help of enzymes is broken down and cells are formed. Then these cells are suspended in a growth medium and they will replicate and they will form a monolayer of cells. So it can be primary, it can be diploid cell line, it can be continuous cell line. Primary cell line example is primary monkey kidney cell line, diploid WI38 and HL8, and continuous HeLa, Hep2, Vero, McCoy and KB.